The Adaptor Hypothesis. This was published in 1958 by Francis Crick. Crick hypothesized that small RNA molecules would act as adapter proteins that can receive a certain amino acid and interact with specific messenger RNA triplet codons via base pairing. He also thought that there would be 20 adapters, one for each amino acid. So, Crick hypothesized the missing link between transcription and translation. The central dogma of DNA replication follows the idea that DNA is replicated, then transcribed into RNA, and then RNA is synthesized and then translated into protein molecules. What's involved in this would be mRNA, tRNA, amino acids, ribosomes, and anticodons. So, mRNA, the messenger RNA, is the copy from transcription carrying the genetic information for protein conversion. The tRNA, the transfer RNA, carries specific amino acids and reads the 3-letter sequence of mRNA. The amino acid is the basis of the proteins, carried by tRNA, which grows into the polypeptide chain. Ribosomes are the protein production center. The units assemble and read mRNA, inviting the tRNA to attach to the correct amino acid, forming the polypeptide chain. Anticodons are the 3-letter base pair on the mRNA, and the tRNA helps transcribe the mRNA code into the polypeptide, and it will move down the chain to create. It will move down the mRNA to create the polypeptide. How is Crick's idea inaccurate? Inaccurate in the idea of there only being 20 different amino acid groups. Accurate that there was tRNA was used. However, he did not hypothesize the rules for the anticodon base pairing when tRNA is being used. Knowing that mRNA was read using tRNA inside of a ribosome started the study of the anticodon base pairing. The anticodon wobble rules show that a single tRNA can pair with more than one codon, which shows how there's 64 possible codon combinations, but only 31 to 41 adapter proteins. If there is an I present in the mRNA, then it cannot code for anything because there is no anticodon that matches with I. So this would create a missing amino acid in the chain, which can completely change the protein shape by changing the way it folds.